CRISPR Cas9, it's a new frontier in infertility treatment, harnessing gene editing for reproductive health. Welcome to our presentation on CRISPR Cas9 and its revolutionary role in infertility treatment. CRISPR Cas9 is a groundbreaking gene editing technology that allows scientists to edit part of a genome by removing, adding, or altering the section of the DNA sequence. Understanding genetics is crucial in fertility, as genetic issue can affect sperms and eggs quality, leading to infertility. Genetic abnormalities can lead to various fertility issues in both men and women. These include chromosomal anomaly and or specific gene mutations. Gene editing is a type of genetic engineering in which a DNA is inserted, deleted, modified, or replaced in the genome of a living organism. CRISPR-Cas9 technology allows for precise editing to the DNA at a specific location. It has transformed genetic research by providing a powerful tool to understand and potentially correct genetic defects. Studies have shown CRISPR-Cas9 could help correct mutation in genes that affect sperm production and function, potentially restoring fertility to a man. CRISPR-Cas9 edit genetic is also being researched. Its potential to correct genetic defects that affect female fertility, such as those impacting egg quality and including implantation. Yes, there are ethical considerations, which is very important. Gene editing in humans, especially embryos, raises significant ethical and legal questions. It is essential to navigate those carefully and consider the long-term impact. Recent development have moved CRISPR from laboratory to clinical trials. In other words, CRISPR-Cas9 edit genetic have actually moved now to work clinically to patients. All the animal studies are completed, and this engineering, the best art or technology, is now ready to be implemented in human beings. However, in 2018, one of the doctors have actually implemented or used this method in human, in which twins were born, and fortunately there was no harm. Although the scientists felt that it was too early to test this modern technology to human. But in this scenario, Crispr Cas9, it was used by this scientist to prevent HIV infection. It is believed that if you can mutate, edit, or remove a certain gene in the DNA, the chances that you can get infected by HIV is non-existent. This is still on research, but the possibility are very high that most likely HIV will be eradicated by Crispr-Cas9 gene editing method. Now, including many other diseases, especially infertility issue, Crispr-Cas9 editing can be used. Potential for Crispr-Cas9 technology in treating infertility is vast, offering hope for cases previously deemed untreatable. While promising, Crispr-Cas9 technology faces challenges such as off-target effect. By off-target, it means you can miss if the problem was in gene number 18. Instead of taking or mutating gene number 18, you went and miss it and go to 19 with disaster. And that is called off-targeting. In other words, you have missed what you wanted to correct. Ethical issue as well on especially gene editing. Some people, they say, I want to be taller. Some I want to be short. Some I want to be intellectual. I want to be genius and all that. Those are ethical issues. And this modern advanced technology is not meant for such. Another ethical issue, it may that people who are financially okay may want to predict or edit their offspring to what they want. Therefore, this method, from ethical point of view, it shouldn't be used for such endeavor. Ensuring long-term safety and effectiveness of Cas9 technology is very important. In conclusion, Cas9 offer exciting possibility for the future of fertility treatment. At Joburg Fertility Center, we are committed to staying at the forefront of this advancement to better serve our patient. Thank you for listening. Thank you for your time.